general area, 3 Antipolo streams. May I call on Marina Nicole Salvador, who will speak on behalf of her team about their nice project. Marina?
Good morning. I'm Marina Nicole Salvador, and I'm from the Holistic Education and Development Center, also known as HEDCEN, and I am here to talk about my group study entitled The Reintroduction of Awos Melanocephalus in 3 Selected Streams in Antipolo. So here is the outline. The introduction. Freshwater fish are very important, and their importance cannot be overstated. However, rates of local extinction remain very high, and some of the causes are overfishing, pollution, and the introduction of alien fish. That is why our group aims to do the following. We first aim to do a water quality assessment of the 3 rivers, and to compare the species richness. Also, of course, to reintroduce the Awos melanocephalus. For our significance, the Awos melanocephalus is an edible fish, so those living around the rivers can catch these fish and they can eat them. Also, through our study, we were able to provide samples for the Institute of Biology and UP Diliman of the Awos melanocephalus, which they used for their DNA barcoding. The Awos melanocephalus can also be used as a bioindicator and to strengthen the trophic structure because it eats gobies, and as well it is eaten by certain birds such as the kingfisher. And also, if there is local extinction, we can use the 3 rivers to get the locally extinct fish as a source of the stocks. So for our equipment, for the water quality assessment, we use the YSI EcoSense meter for the dissolved oxygen, pH, salinity, conductivity, and TDS, and the EcoLight Deluxe light meter for the light. And what we did was a one-time sampling basis. For the flow rate, we had our homemade instruments for flow rate and the transit tape for river width and depth. So for catching the fish, we used these traps. First is the patanga. Next is the bubo, which comes in 2 sizes. One is 4 feet and we have 5 1-foot bubos. And the gill net. For our study areas. So over here, you can see this one is Laguna Bay, and this one is La Mesa Dam. Over here, we have Mangahan Floodway, Marikina River, and Pasig River. So, we're there. Zoomed in image. So, those are the rivers. That one is the Tongtong River, so it empties to Mangahan Floodway. Next is Dalig River, which also empties to Mangahan Floodway, and the Muntingdalao River, which empties to the Dalig River, which goes to Mangahan Floodway. So another zoomed in image. So this one is Antipolo, and here are study areas. So from over here, you can see that our study area of Dalig is very far from the source. This is the source, this is our study area. While for Muntingdalao, this is our study area, and over here is the source, so it's very close. So here you can see that before reaching our study area, the river, Dalig River, first had to cross areas which are accessible to the community, so it is open to being polluted. Also for Tongtong River, it also crosses a short distance where it is open to the public. While for Munting Dalao, its source is almost, its source is inside the subdivision, so it's protected. So, the Munting Dalao River, which is located in Fairmont Hills, here are its parameters. And for the basins, we used to set up our traps. And next is the Tungtung River. This one is the deepest, is the deep basin in Tungtung River. It is around 3 to 4, it is around 4 meters deep. Oh my. Okay. So this is the other basin where you set up the traps. And next one is the Dalig River. And here its parameters. So this one is the deepest, this one is the most polluted river, and here are the photos so you can see. That shows how fast the current is. And you can see the trash. And also it smelled very bad. Maybe that's why it's called Sabang Baho. So for our results, for the water quality, you can see over here that the DO is decreasing from Tungtong to Munting Dalao. While for the total dissolved solids in the conductivity, it is increasing from Tungtong to Dalig.